Hello, Assalamu alaikum. In this video, we will understand about the scavenger receptor. What is the scavenger receptor and what is this? The superfamily of cell surface receptor it is for use the homeostasis and combating the disease like the arteriosclerosis. This is the special example is the macrophage, which that's contain a scavenger receptor, and the ligand will bind the polyanionic ligand, including lipoproteins, apoptotic cells, cholesterol ester, phospholipids, proteoglycans, ferritin, carbohydrates, pathogen-associated molecular pattern, and death-associated molecular pattern. This all ligand is due to the polyanionic mean it is free radical or oxidized lipids, oxidized phospholipids, or oxidized cholesterol. It will be endocytosed to clear this problem and clear the hypercholesteremia problem. The scavenger receptor biology and the rule of these receptors is modified LDL clearance have increased in last 20 years. In arteriosclerotic lesion, the macrophage that express the scavenger receptor on their plasma membrane, and whenever the endothelial dysfunction will lead to invasion of the monocyte after the recruitment due to the inflammation, due to the deposition of the lipids, whenever the lipid in deposition will recruit the monocyte due to the inflammatory mediators, and in this way, the endothelial dysfunction will lead to macrophage in endocyte moving to the tunica intima, where the atherogenesis will occur, low density lipoprotein oxidized, and the macrophage, which that is important to convert this foamy cell after the lading and endocytosing the lipid, the LDL, and to convert the foamy cell and produce cytokines to recruit more macrophage, and death to produce more lipid deposition in the tunica intima, lead to inflammation and arteriosclerosis problem. Anyway, let's understand about the all classes. Class A, first we will discuss. These contain a different type of receptor, like scavenger receptor A1, A2, and A3 are present in the endoplasmic reticulum, while the Marco and scavenger receptor CL is this is the 5 receptor involved. The receptor is expressed mostly in the macrophage, and the macrophage foam cell expression in atherosclerotic plaque, and the scavenger receptor A1 and 2 recognizing the B ApoB protein component of these particles. The gene expression on the basis of their cell. For example, the macrophage produce this gene to produce this type of receptor. Anyhow, the apoptotic cell, the anionic phospholipids and glycation and products is involved for the endocytosing and the sensitization of these things. Pathogen associated molecular patterns, gram negative lipopolysaccharides, and Neisseria meningitis, and the gram positive Staphylococcus aureus, and the Listeria monocytogenes. These all factors is used for the endocytosis of these content for the clearance. But the class B is the scavenger receptor CD38. The both N terminal and carbon terminal is the cytoplasmic domain. And in this way, the scavenger receptor B1 and B2 is involved as well as the CD36. The scavenger receptor is the class B scavenger receptor is like macrophage and contains CD36 molecule as a receptor, and the oxidized lipid low density lipoprotein will endocytose for the clearing and decreasing the reactive oxidative stress. Normal and pathological process is the apoptotic cell clearance, fatty acid transport, adhesion, angiogenesis, atherosclerosis, Alzheimer's disease, and microbial defense. Anyhow, These class show this receptor on the monocyte, macrophage, adipocyte, the fat deposit cell, microvascular endothelium, and dendritic cell. The parasitized erythrocytes is identified the phospholipids apoptotic and thrombospondine and plasmodium falciparum is detect and the CD36 receptor containing these erythrocytes. In this way, the myeloid stem cell also contain a myeloid cell contain basically are present in the bone marrow also contain this Scavenger receptor. The Scavenger receptor B1 and B2 is whenever decrease on the surface of the cells of the macrophage will lead to hypercholesteremia. The express in liver and macrophage. Lipoprotein metabolism involved in two ways. Number one is the Scavenger receptor B1 mediates the cholesterol transfer from cell to HDL, while on the other hand, the facilitates the selective recovery of these cholesterol from HDL to stereogenic tissue and to the liver for excretion into bile and feces. We are not understanding about the diagram and how it can work, but just overview. You can identify and this check through the other research ideas to understand like googling. This let's here is the class E contain oxidized low density lipoprotein identification and recognition to endocytose and the intramyocardial vasculopathy pathological effect. Class E is the expressed in macrophage and smooth muscle cells, and this contains this like a receptor and lectin like. It is lectin like. And type 2 transmembrane protein that was identified as a receptor for oxidized low density lipoprotein in bovine aortic endothelial cell and advanced glycation and products apoptotic cells and bacteria is the ligand, while on the other hand the present in the atherosclerotic lesion of humans and hyperlipidemia in mice. While the class F is contains the Scavenger receptor EC1 and 2. This class D contains transmembrane receptor containing N-terminal epidermal growth factor like domain trans A and the transmembrane domain minor role in foam cell for cell bind with modified LDL. Only Scavenger receptor EC1 internalized and these ligands for degradation 6% of total acetylated LDL degradation. So anyhow, in this way you can understand this is the basically is the transmembrane domain and this minor role in foam cell but bind with the modified LDL and only SRIC1 internalized this ligand for degradation of total acetylated LDL degradation. So basically it is used for the LDL degradation. So remember, on the other hand, the class D Scavenger receptor is the CD68 and macrocyaline. This macrocyaline. It is CD68 and it is a murine homology. The macrocyaline are heavily glycosylated type 1 transmembrane proteins that are predominantly expressed in late endosome and lysosome of macrophage. Receptor expressed in macrophage foam cell in atherosclerotic plaque of ApoE. Anyhow, this is a class G. This class G is contain a Scavenger receptor ROX and the CXCL16. This whenever the oxidized low density lipoprotein will bind with the Scavenger receptor in this the class G basically is the present in smooth muscle, endothelium, macrophage, CXCL16 gene polymorphism with severity of coronary stenosis, Scavenger receptor PXOX was identified by its ability to bind the oxidized low density lipoprotein and was subsequently shown to be identical to the membrane bound CXC chemokine CXCL16 and expressed in human and mouse atherosclerotic lesion. So anyhow, this is the basically the class G contain a Scavenger receptor ROX and the CXCL16, and this contain two domains, remember. So anyhow, it is important for the homeostasis and combating the disease, and the macrophage basically involved for the cytokines production after the endocytose the ligand will converting the foam cell if this is a cholesterol. If the cholesterol, the LDL will be deposited in the macrophage, and in this the pathogen associated molecular pattern which that will also involve. In this way the atheroma formation, the chronic inflammation, thrombosis, etc. will be occur. But this is the clearance mechanism for short. If the endothelial cell will be dysfunction, that will lead to cause atherosclerosis. Otherwise, no any problem because the Scavenger receptor is important for the clearance of the cholesterol. But whenever the endothelial cell dysfunction due to the type 2 diabetes mellitus will lead to deposit into the tunica intima. Anyhow, I hope you make sense about this. If you like my video, please make sure to subscribe, like, and share. This is the video was about the Scavenger receptor, superfamily of cell surface receptor overview. I hope you make sense about that. Basically, the Scavenger receptor is used for the clearance of the lipids and other pathogens and other type of the ligands, polyanionic ligands, and in this way the disease inhibition and immune system. Thanks for watching. Bye.